Hello, everyone. I'm Sachleen Tuteja, and I greatly appreciate this opportunity to present my research work, where I evaluated the performance of variant annotation tools used in the clinical bioinformatics pipeline for genomic testing. So since its inception in 1959, when Down syndrome was shown to be caused by an abnormal number of chromosomes, genetic testing became well established in the second half of the 20th century, and by the end of the century, around 1,000 of the estimated 7,000 single gene inherited diseases had been characterized, including Huntington's and cystic fibrosis. So, with the completion of the draft sequence of the human genome in 2001 and emergence of transformative technologies in all areas of genomics, the first 2 decades of the 21st century have witnessed a dramatic transition from low resolution single locus genetic testing to high resolution genome wide testing in medical practice. And while single gene genetic testing has primarily been used for risk prediction, disease diagnosis, and assessing carrier status, genomic analysis is enabling additional functions such as disease prognosis and therapeutic decision-making. So next generation sequencing, or NGS, offers multiple approaches for the investigation of the human genome, including sequencing of the whole genome, exome, transcriptome, or a specific panel of genes in a matter of hours to days. So exome sequencing results in the identification of about 20,000 variants, and after systematic filtering for rate and protein changing variants, a single exome harbors anywhere from 100 to 200 potential disease-causing changes. Thus, finding a disease-causing variant among these many genetic changes mirrors the proverbial search for the needle in a haystack. So genetic variants can be grouped into 3 categories. So single nucleotide variations and insertions or deletions that represent changes in the sequence of the genome, and tandem duplications, copy number variations, translocations, inversions, etc., that are changes in the structure of the genome. So the onslaught of genome-wide data has led to the development of a battery of commercial or lab-developed tools for the identification of variants. However, very few variant callers are versatile enough to call all 3 types of genetic variants. So it's often beneficial to incorporate multiple independent tools in the clinical pipeline. So given the wide variety of variant annotation software that are available today for clinical NGS, in this study, I evaluated the performance of 3 variant annotation software. So Ensemble's open source variant effect predictor, or VEP, Annovar, and a commercial tool called Alamut batch. So all 3 platforms generate transcript and protein-based variant nomenclature from genomic coordinates as per HUVS standards. So a test set of 298 intronic and exonic variants across 191 genes previously classified and reviewed in clinical reports by the Molecular Diagnostics Lab at Lurie Children's Hospital was curated and used for the study. So these variants were generated by targeted gene panel sequencing using the approximately 4,700 gene medical exome panel on 105 patients. So the 298 variants were manually reviewed using Alamut Visual and Integrated Genome Viewer, or IGV, and orthogonally confirmed with Sanger sequencing. So after the 2-tiered validation processes, the 298 variants were distributed with respect to their classification. So SNVs, deletions, duplications, complex, or insertions. And these constitute the ground truth set. So as shown on this pie chart, our evaluation revealed that a majority of the variants, that is 276 of the 298, were single nucleotide variants or SNVs, followed by deletions, duplications, complex, and insertions. So individually, Alamut batch, Annovar, and VEP exhibited similar annotation profiles with regards to the distribution of variant types. Although VEP was unable to annotate one of the variants, and Annovar was unable to identify 4 changes. So as depicted on this Venn diagram, a total of 278 of the 298 variants were found to be concordant between the 3 tools. And while Alamut batch and VEP exhibited comparable accuracy and precision with the ground truth, Annovar exhibited the greatest number of discrepancies. And a majority of these discrepancies were non-SNVs, so 13 out of the 20. And of these 20 discrepancies, 16 were exonic, while 4 were intronic. And they exhibited a spectrum of pathogenicity from likely benign to pathogenic. So details on the 3 discordant variants between VEP and Alamut batch are illustrated in this table here. So of the 3 discordant SNVs, Alamut batch called one variant in the CSF3R gene as exonic instead of it being intronic as indicated by the ground truth. So this difference in region can potentially be severe, resulting in an incorrect diagnosis of the patient's condition. So based on this data, it's evident that with respect to the HGVS nomenclature standards and clinical integrity, VEP has produced more accurate genetic annotations. So for the discordant variants, these differences were due to the various transcripts used by the software, which affected their genomic locations and thus produced discrepant results. So to my knowledge, this study is the first relative performance evaluation of Annovar, Alamut batch, and VEP, although similar studies comparing other software packages have been reported. So in conclusion, without accurate genetic variant identification and annotation, clinical interpretation of variants of unknown significance will continue to be a challenge for clinical diagnostics. Therefore, appropriate and validated methods of variant annotations are critical in the post-analytical phases of clinical testing, and in ensuring that patients receive an accurate molecular diagnosis for their genetic conditions. So thank you so much for your time and attention. This investigation would not have been possible without the encouraging guidance of my mentor, Dr. Kylie Yap. And I would also like to acknowledge the support of the scientific inquiry and research team at IMSA. Thank you.